Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Today we are going to discuss one point from immunology, the antimicrobial and cytotoxic activities of macrophage during phagocytosis. So macrophages are basically the immune cells. These are synthesized in bone marrow that circulates in peripheral blood for 8 hours, and then these macrophages resides in a tissue. The main function of macrophage is phagocytosis that engulfs the infectious organism. It may be a bacteria. So after engulfing, it acts upon that engulfed microorganism. It carry out the cytotoxic activity and killing activity inside it, and then it presents that antigen to the T helper cells. So during this phagocytosis, macrophages produces different killing and cytotoxic substances. So we are going to see what is the mechanism of this antimicrobial and cytotoxic activity of the macrophages. Now this activity is divided into two types. One is a oxygen dependent killing mechanism and another is a oxygen independent killing mechanism. Now starting with the oxygen dependent killing mechanism, there can be a 3 to 4 ways by which macrophage can do killing activity. In these ways, basically macrophage synthesizes reactive oxygen intermediates or reactive nitrogen intermediates in it. Both the substances have potent antimicrobial activities. So let's understand more about oxygen dependent killing mechanism of the macrophages. Now in case of activated macrophages during the phagocytosis, respiratory burst occurs. What is meant by respiratory burst? So this is nothing but oxidative burst, or there is a release of reactive oxygen species in a cell, that we call it as a respiratory burst. So when this occurs, there is an activation of membrane bound oxidases in a macrophage. These oxidizes oxygen and it forms the superoxide anion. This is a reactive oxygen species and this is very harmful for the ingested microorganism. This also generates other powerful oxidizing agents like hydrogen peroxide and hydroxyl radicals. In the second way of action in oxygen dependent mechanism, after the phagocytosis, when the lysosome fuses with phagosome, it forms a phagolysosome and there is an occurrence of myeloperoxides. Now this enzyme converts hydrogen peroxide and chloride ions into a hypochlorite. And this hypochlorite is again very toxic to the ingested microorganism. Third way of the antimicrobial activity of macrophage includes activation of macrophage by some cell wall component of the bacteria. In case of normal range of bacteria, it may be a lipopolysaccharide. LPS, that is a bacterial cell wall component, it may activate the macrophage, or muramyl dipeptide, which is from the mycobacterium. If there is a mycobacterium infection, the muramyl dipeptide activates the macrophage. And what happens here? There is a formation of reactive nitrogen species. So along with these components, there is a involvement of T cell derived cytokines interferon gamma are involved in this reaction, we can say, and there is an expression of nitric oxide synthetase. This nitric oxide synthetase converts L-arginine along with oxygen, as this is an oxygen dependent mechanism. So oxygen is involved, NADPH is there, and it forms the nitric oxide. Okay, so this nitric oxide is again a reactive nitrogen species and it is very harmful to the bacteria. Along with this nitric oxide, there is a formation of L-citrulline and NADP is formed. This nitric oxide formed in this reaction and the superoxide anions, which are formed in a first type of mechanism we have studied. If these two combines, these two have more potent antimicrobial activity. Now discussing with the oxygen independent mechanism of the macrophages. See here, there is a synthesis of various enzymes. When there is a combination of lysosome and phagosome, it forms a phagolysosome. So from the lysosome, there is a secretion of lysozymes and other hydrolytic enzymes. So these enzymes itself have a killing activity, degradative activities, and these do not require oxygen for their activities. So this is a oxygen independent mechanism of the macrophage. Secondly, this includes tumor necrosis factor alpha. This is secreted by the activated macrophages and this is cytotoxic for the tumor cells. Third oxygen independent mechanism includes involvement of the defensing. So these are antimicrobial and cytotoxic peptide. This is a protein. It is rich in the amino acid cysteine. Now, due to the presence of cysteine in a defensine, it can form a disulfide bond. And using disulfide bond, this defensine forms ion permeable channel in a bacterial cell membrane. So due to this, bacterial killing can occur and it can have an antimicrobial effect. This defensine is activated against the Staphylococcus aureus, Streptococcus pneumoniae, Escherichia coli, Pseudomonas aeruginosa and Haemophilus influenza. So these bacteria can get killed by the formation of ion channels using defensine in their cell membranes. So here I have concluded with today's video. I request you to please like, share and subscribe for my channel and also to hit the bell icon. Thanks for watching.